Welcome to Epigenetics3, Histone Modification and Chromatin Immunoprecipitation. In this course, we will explore how gene expression is controlled by histones and how histone modification can be studied using specialized protocols like chromatin immunoprecipitation. We will also discuss analytical approaches to study genome-wide histone modification data, or ChIP-seq, and specific challenges these methods are designed to address. Before we jump in, let's first do a quick overview of epigenetic regulation and discuss the significance of histone modification. As you remember from previous courses, epigenetics is the study of mechanisms that cause changes in gene expression, but that are not encoded in the DNA sequence itself. These mechanisms include DNA methylation, histone modification, activity of non-coding RNAs, such as microRNAs, and the regulatory function of non-coding repeating regions found in the DNA. Histone modification is a major aspect of epigenetic regulation. Histones are proteins that the double-stranded DNA is wrapped around. On these images, you can see how the DNA string has small bumps or beads on it. These visible beads are nucleosomes that are made up of histones. When these beads were originally discovered, scientists confused them with genes until it was eventually proven that the string-like structures wrapped around, and not the beads themselves, actually contain the genetic code. Histones can be either grouped together, forming lumps of DNA, or as we can see in the picture on the right, more relaxed and spread out. It turns out that the modification of histones plays a major role in how densely the histones are grouped together or spread apart. Moreover, additional modifications of histone tails can have other effects on gene transcription. One important aspect of histones is that they can be changed to alter how much packing the DNA is capable of. There are several key modifications that take place as a result of changes to groups of atoms that are at the ends of histones. These changes can be of several types and will have a positive or negative charge that either attract them closer together or force them apart. As a result, the DNA region that is wrapped around the histones can be more or less accessible, causing variation in gene expression. The nucleosome is considered to be the basic repeating unit of chromatin, in which 146 base pairs of DNA are wrapped around an octamer of core histones consisting of pairs of H3, H4, H2A, and H2B. N-terminal tails of these histones protrude out of the nucleosome and are subject to a variety of post-translational modifications such as acetylation, phosphorylation, ubiquitination, and lysine and arginine methylation. Acetylation was the first of these modifications to be linked with active transcription, and subsequently, phosphorylation of histone H3 was found to cooperate with acetylation in transcriptional activation. Some histone methylation events have also been associated with transcription activation and others with gene silencing. Histone modifications can function either individually or combinatorially to govern such processes as transcription, replication, DNA repair, and apoptosis. It has been proposed that methyl group of atoms increases packing and acetyl group decreases packing. Also, phosphoryl group can be attached to the histones and causes a decrease in packing. These modifications and their impact on transcription and other processes are being actively studied, and as our understanding grows, researchers appreciate how complex this machinery works in real life. As we mentioned, histones are organized in octamers, forming nucleosomes. The four histones are called H2A, H2B, H3, and H4. DNA is wrapped around the structure 4 times with histone H1 holding everything together. The most studied modification of histones are methylation and acetylation of the core histone tails. As you can see, modification to the tails of histones is linked to the length of the tail itself. When a specific type of modification is studied, it will be referred to as H3K4-ME3 or H3K27-ME3. The name simply refers to the position on histone tail and the type of modification it is. Here you can see a table of histone modifications and their reported impact on transcription. For example, you might read about H3K4 methylation-free signal. This means that H3 histone tail is being modified by removal of methyl group atoms. Different signals act as different regulatory mechanisms and sometimes work together in complex patterns. The function of specific histone modifications is an active field of research, and many of these modifications are not yet known. On the right, you can see how these modifications appear in ChIP-seq data against the control sample. To understand this better, let's discuss chromatin immunoprecipitation for studying histone modification. Chromatin immunoprecipitation followed by high-throughput sequencing, or ChIP-seq, is a powerful tool to identify genome-wide profiles of transcription factor binding sites, histone modifications, and nucleosome positioning. To understand the way this data is generated, we have to look at the process in greater detail and appreciate the biological elements involved in various steps of the ChIP-seq protocol. Chromatin immunoprecipitation uses antibodies designed to bind to specific proteins of interest, or POIs. These can be histones or other complexes attached to the DNA. First, cross-linking ensures the proteins are tightly bound to DNA so that DNA shattering does not remove them. DNA is shattered, leaving fragments of DNA with or without protein of interest on them. Special antibodies bind to fragments of DNA with proteins of interest. The antibodies are typically attached to magnetic beads so they can be used to extract fragments of DNA with protein antibody complexes. The complexes can then be removed and libraries for sequencing are prepared. The sequenced reads will contain tags that are used to denote the position next to where the protein was bound to DNA. As a result, peaks of reads will accumulate next to positions of DNA bound to protein. In the analysis step, these positions are identified, peaks quantified, and position of the protein binding site is determined. Accumulation of reads with tags is analyzed for accurate detection of binding sites. To detect accumulation properly, a second library called iDNA is prepared as a control. Abundance of reads versus control provides assessment of abundance significance. Then, reads aligned to positive and negative strands are analyzed for overlap, helping select the exact position of POI binding site. A key step for chromatin immunoprecipitation is the design of an antibody that binds to a specific protein. For example, you might have a kit for histone H3, or even more specific to H3K27 or H3K4. The specificity of the antibody, whether it is monoclonal or polyclonal, and the organism it is specific for are all key factors in generating high-quality data. The whole process of data preparation looks like this. Proteins of interest, like histones, are bound to DNA. DNA is fragmented, resulting in DNA fragments that have protein of interest on them and others that do not. They might have other proteins on them or nothing at all. Only DNA with proteins of interest bound to them are of interest. They are selected with specially designed antibodies. The DNA is then separated from the protein and the DNA fragments sequenced. As a result, reads that were in the region bound to the protein of interest can be aligned to the reference genome, and regions with abundance of reads are analyzed. When we deal with ChIP-seq data, we are analyzing sequences of short reads that came from DNA fragments that were attached to protein of interest we selected. After we sequence the raw reads, they need to be cleaned from adapter sequences and PCR duplicates. Then, two, their cleaned reads are aligned to the positive and negative strands of the DNA separately. This is done because when the DNA is wrapped around the protein of interest and fragmented, there will be a slight shift to the left or right, left from the positive strand and right from the negative strand, that needs to be accounted for. Then, the challenge is to accurately identify peaks, what is commonly referred to as peak calling. Peak calling is performed on the two strands separately, which leads to the next step, determining the position of the POI center by shifting the positive and negative strand reads and identifying the intersecting segments. Finally, the intersecting segments are annotated and marked as positions where the DNA was wrapped around a POI. The major objective of ChIP-seq analysis is to detect genome fragments that are enriched with upregulated signals. These fragments could be transcription factor binding sites, chromatin remodeling, or gene transcription events. The main algorithmic challenge is to accurately detect enriched genome fragments, generating a whole genome landscape for the signal. To do that, let's review several techniques related to peak detection and peak shift that are a part of the ChIP-seq workflow. Probably the most discussed issue in ChIP-seq experiments is the best method to find true peaks in the data and precisely identify a binding site. A peak is a site where multiple reads have been mapped to and produce a pileup. To accurately assess the pileup significance, a control library is often used, also referred to as iDNA. ChIP sequencing is most often performed with single-end reads, and ChIP fragments are sequenced from their 5' ends only. This creates two distinct peaks, one on each strand with the binding site falling in the middle of these peaks. The distance from the middle of the peaks to the binding site is often referred to as the shift. As the DNA is wrapped around the histones and can be more or less accessible, various portions of the DNA are affected. These include the promoter region, transcription start sites, and the introns and exons of genes. Other regulatory regions, like enhancers and microRNAs, are encoded in introns or in proximity to coding regions. Each one of these elements can have a significant effect on the level of gene expression, alternative splicing, as well as other downstream regulation of pathways. The size of the region and its location are both important. Researchers are reporting that different histone modifications are associated with specific widths and profiles of peaks that can be detected in ChIP-seq data. Oftentimes, specific regions like enhancers and transcription factors or microRNAs are being studied. In cancer, neurodegenerative diseases, and other conditions, researchers are studying one or multiple types of modifications and the elements they regulate by comparing ChIP-seq profiles of groups of samples. Accurate identification of differential profiles of histone modification can lead to the understanding of epigenetic regulation specific to a condition. Now that we understand the way ChIP-seq data is prepared and analyzed, let us try to perform an analysis ourselves. In this hands-on section, we will build a simple pipeline to analyze the data and discuss methods that were used and pipeline results. Before we jump in to building our pipeline, let's review the options we have for analysis. The two options are for analysis of peaks in a given sample or for detecting differences between two conditions. In our case, we will use the iDNA option to identify areas that have histone modifications by comparing a condition versus control. The condition we will study is Angelman syndrome, an inherited neurodegenerative disease. Angelman syndrome, also known as AS, is a neurogenetic disorder caused by deletion of the maternally inherited UBE3A allele and is characterized by the development delay, intellectual disability, ataxia, seizures, and a happy effect. It is believed to be caused by epigenetic deregulation of a region that is responsible for proper neuron differentiation. We will use a pair of samples from the study to try a hands-on analysis of ChIP-seq data. The full article is available here. Let's navigate to server.t-bio.info and open the areas of analysis. Here you will see a dedicated section for analysis of ChIP on ChIP and ChIP-seq data. When you are in, select single-end reads, SE, and Homo sapiens GRCH38 for reference genome. We already have two FASTQ files in SRA format, archive of sequence reads, uploaded to the server, so we don't have to wait for lengthy uploads over various connection speeds. SRR 5990885 is a healthy control sample, and SRR 5990882 is a cell line from a patient with Angelman syndrome. We can place the healthy in sample group and sick in iDNA. Essentially, we will get a contrast between the two, so it does not matter which one goes where. Our first pipeline will be very simple. For now, let's skip the pre-processing steps and discuss two central processing steps, alignment and segmentation. Alignment, or mapping as it is also called, can be done with several methods. The main goal is to align reads to the reference genome we selected, Homo sapiens GRCH38. Bowtie2G is a newer version of the Bowtie algorithm that was published in 2012. The second main step in the analysis of map reads is accurate detection of enriched regions. For this purpose, we will use iSeq, a method that uses a Bayesian hidden icing model for ChIP-seq data to ignore falsely enriched regions caused by sequencing and/or mapping errors. Another, more complete pipeline is shown here. The main change is with including PCR clean to remove PCR duplicates and using multiple methods for segmentation. These include BIN-S, a segmentation method developed by the Taiwan Bioinformatics Research Center, and MAX2, a popular method for analysis of ChIP-seq data. BIN-S algorithm is based on a method developed by Brodsky et al. In 2010, called the binary search approach to whole genome data analysis. This divide and conquer type algorithm detects a series of non-interesting fragments of various lengths with locally optimal scores. Original version of MAX2 was published by Ying Zhang and Tao Liu from the lab of X. Shirley Liu at the Dana-Farber Cancer Institute, Boston. Here we will use version 2, developed and maintained by Tao Liu. As a result of the second pipeline, we can use mapping statistics as well as a table of each chromosome. Let's select chromosome 1 to see what the output looks like. In the output table, you will see a number of columns corresponding to outputs by each method: BinS, ChIP-seq, and MAX2. The most reliable results will be when each method gives an enrichment score significantly greater than 0. For example, these positions highlighted between 961,200 and 961,800 on chromosome 1. Since the output is already annotated, we can see this segment corresponds with ANSG 00000187961. This is KLH17, Ketch-like family member 17. Let's look at this gene in greater detail. The protein encoded by this gene is expressed in neurons of most regions of the brain. It contains an N-terminal BTB domain, which mediates dimerization of the protein, and a C-terminal Kelch domain, which mediates binding to F-actin. This protein may play a key role in the regulation of actin-based neuronal function. KLH17 has been reported to be involved in neural development. For example, in their study of infantile spasms, ISS, Pakyurakovsky et al. Linked KLH17 to expansion of epilepsy phenotypes. As you can see in this chart, where enrichment scores from ChIP-seq and MAX were plotted, KLH17 is only one of the genes identified by enrichment of reads. As you remember, these reads mark a protein of interest attached to the DNA. Reading deeper into the method section of the paper, we can find what antibody they used for preparing their ChIP-seq data. The protein of interest was H3K4-ME3. If we plot the scores on a scatter plot with y-axis in the position on the chromosome, we will see that KLH17 is at the start of the chromosome, one of the first enriched regions identified by all three algorithms. But that is not the only region that we can see highly enriched in this type of methylation of H3 histone. We also see another region, around the 150 million position, that stands out. One of the ways we can gauge the significance of a whole region is to use David for enrichment with gene ontology terms. Now that you have this data, you are welcome to play around with these enriched regions to see what you can discover. Thank you for taking this course, and we hope that you enjoyed learning about epigenomics data analysis. If you want to take things further, we recommend diving deeper into the Engelman project or this other project focused on asthma. To help you get started with these datasets, we prepared guides on accessing all of the data that include ChIP-seq, bisulfite-seq, and RNA-seq datasets. In the project, you will find data, guides, and explanations of methods you can rely on analyzing and integrating this multi-omics data.